Thank you, Nicolas, for the kind introduction and for putting pressure on me on the content and on the timing of my talk. So thank you also though, for creating this possible. How was the last time you did something for the first time? Coming to my age, it becomes more rare. It's not something happening every day, every week. But then when I come back to France, I will say that this week I had the chance to go for the first time to Oregon and to Napa. Coming back also to catching up with friends at Scott Labs, so really, and sharing times with Pepe. And so thank you for that opportunity. Well, I will talk to you about specific fungal chitosans, one of my favorite topics for over than 15 years now. So this again makes me remember that I'm not very young anymore, I'm just young. And so we've been working at L'Allemand within the group for 15 years, a bit more than 15 years actually, in partnership with a company based in Europe producing these chitosans from Aspergillus niger. And yes, the purpose of sharing this experience and know-how with you today is just to make clear that it's not one chitosan, actually it's chitosans. And so what I will try to explain to you is why this molecule is diverse and how can it be that you can see many formulations, many preparations that are offered to you as tools in winemaking for different purposes. Outside the biocontrol, antimicrobial action, you know that there are many other preparations that can be offered to protect wines against oxidation, fine, clarify, and so on. But we don't talk about the same raw materials of chitosans, and most often these products are formulated with other components that bring synergies and interest to that. So chitosan is derived from chitin. Chitin is the second most spread polymer on Earth, also a polysaccharide like cellulose. Why? Because it's part of the exoskeletons of crustaceans and also part of the cell walls of certain fungi like Aspergillus niger. But not only, also mushrooms, we can find chitin, also in yeast we can find some chitin, not as much as in Aspergillus niger, but we find that. And so chitin glucane, and other molecules derived from chitin have known many applications in the different industries. But recently, well, not recently, because now we have 15 years of experience since it was admitted by OIV and European Union. So back to Europe, we have had the opportunity to make experiences of usage of these molecules, but you have to pay attention that it's only chitosan from fungal origin that is authorized and not crustaceans. However, if you go to pharmacies, you will find a whole bunch of offers of dietary complements and so on, but generally this chitosan is produced from crustaceous exoskeletons. So only one origin is authorized here, specifically in the US, is Aspergillus niger. In Europe, we have two, Aspergillus niger and Agaricus bisporus. But I will come back to that. So here, just to illustrate, I won't enter into the detail, but you can find as many references in the scientific literatures, also many products, commercial products that are used on an industrial basis. For water treatment, for detoxifying, for antimicrobial action protections of grains in agriculture, in food. Also, not only for antimicrobial properties, but also for textural interest. So coming to regulatory aspects, just one slide. It's not my favorite topic, but it's something that we need to pay attention to. Of course, you have, but also us, we have the responsibility to offer you products that comply with the regulatory. And of course, in Europe, we talk about OIV, and you know that the Enological Codex has pages and pages of specifications for the product. And here, as far as US is concerned, the products we offer, chitosan and chitin glucan, which enter in one of the preparations, are complying with the TTB regulations. So what are they? Where do they come from? As I said, we talk, it's extracted from Aspergillus niger, but not any part of this fungi. It's extracted from the mycelium of this fungi, which is rich in chitin and chitin glucan. So you have here a schema illustrating how we can derivate the chitosan on one side and chitin glucan on the other side. I will talk mainly about chitosan today. Chitosan is derivated from the chitin, through that the chitin is extracted from the mycelium and purified in order to have the most pure chitin that we can, and then apply a chemical reaction that will provoke a deacetylation of the molecule. Maybe the figure is small for you, but I will come back to that. You see that you have some acetylated group on the NH function of each unit of the molecule. And when we talk about deacetylation, it consists in getting rid of this acetylated group, and this brings charges. I will come back to that. So for chitosan is a very neat molecule, macromolecule, very interesting because it can vary in terms of chemical properties depending on the size of the molecule and the level of deacetylation that provide different properties of charge to the molecule. On the other side, we can also extract the copolymer, chitin glucan, that is also approved for winemaking applications. Has some clarification properties that actually we found interesting in combination with chitosan for one of our preparation. Main interest of this is that we talk about a biological solution, and that is non-allergenic. On the contrary to chitosan that would come from crustaceous. And so that's why, because of this allergenic risk of chitosan that could come from other sources than the one of fungi, that the experts at OIV put a lot of attention at the right specifications that should be respected to ensure that it comes from fungi and not crustaceous. So here you have the three main characteristics, and one of them being the residual glucans. Glucan is present in the cell wall of fungi. It is not present in the exoskeleton of crustaceous. It's very something that is specific. The other parameters also are a good marker of the source, of the fungal source. But you see out there are very few preparations that are pure chitosan. They are most often blended with other components, sometimes inactivated yeast, cell walls, which brings glucans. And so this brings now a question about the risk of finding this crustacean origin in the preparation that are offered on the market of winemaking. So that's why there are some more sophisticated analyses that are developed by some researchers, both in Belgium and in Italy, like this paper is reporting, of the combination of three techniques, one being very sophisticated, that's the specialty of Fondazione Edmund Mack in San Michele d'Aladige in Italy. Matteo Perini is the fraud lab for the detection of adulteration of food in the Italian market. And so they've been developing a method based on stable isotopic ratio that on pure product can really robustly, reliably demonstrate whether it's fungal or crustacean. And there are two other techniques that are more simple: thermogrammetry, infrared. This rings a bell to you. Infrared is something very common also. With some adaptation of this method now, it can bring a reliable tool to decipher. So thought it would be interesting to share that with you because it's our responsibility to share the knowledge about this, and so that you know that there are these techniques that are available now. Now, coming back to us as a group within L'Allemand Enology, we've put efforts into these fungal chitosans because since the beginning it was exhibiting very nice potentialities for the winemaking process. The first one being the clear first experiments that had shown the strong impact on Brettanomyces. Brettanomyces is not a new topic, as my friend Pepe was saying. It keeps us animated. And so, again, at this time, we found out that chitosan might be interesting. So we were working with our partner, trying to find the best raw material, and we came up with Nobret inside. That is a pure 100% chitosan preparation, micronized, and so with a low granulometry. And then we developed, step by step, we observed that depending on the microorganisms that we target, one or the other of the raw material, you will understand what can be the differences in between different chitosans, could be more efficient to target lactic acid bacteria, acetic acid bacteria, or yeast or so, and also in combination with other components. So that is our story. That is why we are really addressing specific chitosan solution depending on the target you have. And even if we consider outside the antimicrobial effect, because within our group, as you know, we are also proposing preparations for fining, for clarification of must, of wine, for anti-radical activity, and so on. Chitosan is a very versatile molecule that can have many properties, but why can there be so many different preparations outside there that are proposed to you? So I come back to what I was saying about the deacetylation level of the molecule. So you recognize the polymer of chitosan, and circled in red you have this amine function, NH2. And in the conditions of the wine matrix, wine pH, this is positively charged, NH3+. And so actually we don't have a polymer as such. It's a polyglucosamine. Glucosamine is charged, and so that explains how chitosan can interact with so many things in your must or in your wine. Can be microbes, can be polyphenols, can be other compounds, maybe. We are still investigating on this, but always screening different raw materials, because it's not just one. We have different polymerization grades. The number of units makes it a smaller, medium, or longer polymer. So the size, the charge also, we can play in between 10 to 30% of deacetylation, for instance. So you can imagine it will act on the charge of the molecule and so on its ability to be active for the purpose. And finally, when we are talking about powder products, such as the ones we are proposing for antimicrobial action, we can also play at the drying step on the granulometry of our chitosan. And I was saying that actually Nobret inside is micronized, so there are very fine particles that are, it is an important point to explain why it is the most efficient one to struggle Brett. Now, if I come to the action mode of the molecule, we will, from now, just talk about antimicrobial action and opportunities with these preparations. So antimicrobial action is reported a lot in the scientific literature because crustacean chitosan has animated lots of industries, lots of academics on the topic. And so there you have the different applications that are known. But we can find in this literature that is very wide, there are hundreds of different species of microorganisms that were addressed in the different papers. So you have some references at the bottom of the slide. But if we make an extract synthesis of what is reported, we can have a clear understanding of how the molecule can act. These are hypotheses, general hypotheses, but some of the paper will prove that actually this is likely to happen. So on the schema, you have a cell that is represented with its cell wall, plasmic membrane, and intracellular medium. So remember, chitosan is charged positively. One of the barriers of the cell is the lipidic membrane. Lipids are negatively charged. So electrostatic interactions may occur and disturb the balance, the architecture, the organization of the cell membrane, and so provoke a permeability increase that leads to the death of the cell. Other point, remember it's a polymer, can be a very big molecule, so it can also stick on the cell wall and so obstruct the exchanges in between intracellular and extracellular medium. It was also proved in some of the paper for certain type of bacteria, that there are specific interactions with molecules that are present in the cytoplasm of the microorganisms. Firstly, proteins, namely enzymes. You know how they are important for microorganisms' metabolism, but also DNA, RNA. So we can easily understand that all this will not help the microbes. They are struggling and they die. There's really a biological effect. And one last explanation is also due to the capacity of chitosan to trap metals, to combine with metals. And so in that case, metals are part of the micronutrients that are essential for microorganisms. And so it also participates to the inhibition, the decline of the population by enhancing deficiencies in these micronutrients. But if we come now to our world of must and wine and how to struggle against Brettanomyces, that was our first target with no Brett inside, I will share with you some pictures from works that are not very recent for some of them, and some that are more recent, because we need to always stay in alert with the evolution of the techniques. So if we started with counting on plates 15 years ago, quantitative PCR, now there are flow cytometry that is developing and other techniques, so we continue continuously to challenge our solutions with the techniques that appear. But yeah, simple, well, microscopy, but electron microscopy. Interesting, because we can go closer to what is happening there. You have on the first picture on your left, Brettanomyces cells in a wine matrix that look in a good shape. They are not bothered at all. On the two other pictures on the right, you have the same wine with the same contamination of Brett, but with an addition of no Brett inside at 4 grams per hectoliter. And you can see the direct interaction between chitosan particles and how also it impacts the aspect of the surface of the cell. You can observe some wrinkles, that correct? And so it's not, the surface of the cell is not that smooth. And so it's something that corroborates what was reported in the literature. Something is happening there. Yes, there is a direct interaction, physical interaction, that is provoking a change in the cell wall aspect and so on. This was done with Dr. Charlie Edwards in Washington State University. We also worked with a team in Toulouse, where I come from, and here, well, optical microscopy, pretty basic. Very interesting to have one in the winery. This is something typically that you can observe when chitosan is in contact with a wine contaminated with Brett. If you adjust the sample correctly and so on, and have a coloration with methylene blue, you can observe two important things. First, aggregates are formed when chitosan is in contact with Brettanomyces. And secondly, you can see that many of the cells are colored in blue, meaning they are dying, or they are dead. And then electron microscopy, also with the equipment they have in that team, could highlight really the direct contact of the cells with the chitosan particles. Just, well, several of these kind of picture that really comforted us in the idea that it's not only a fining effect that we have when we add chitosan in a wine contaminated with Brett. It's really, there is a biological effect also that causes the death of the cell. Talking about advances in the analytical method, here are some pictures of epifluorescence assisted with some markers of mortality or viability, red meaning dead and green meaning alive. On the left, the control untreated, and on the right, the same wine contaminated but treated with our preparation. And illustrating the loss, the increase of the permeability of the cells, it was interesting to go to this approach by measuring the ATP flow. So of course it's lab scale experiment. This takes place in small scale in a model medium, simulating wine conditions, which is inoculated with Brett. And we either leave it as such, is the control, that's the blue line, or we treat with no Brett inside, in that case at 10 grams per hectoliter. And we would measure in each of the modality the ATP concentration in the extracellular medium. You can imagine that ATP is an energetic compound that is important for any living cells, especially for these microorganisms. And so if it leaks outside, it means that we have disruption in the organization of the membrane that loses its étanchéité. And so you can see clearly orange line increase in the medium. So this really confirms the biological lethal effect of the chitosan. And another point interesting here, maybe it can be small from my eyes already, so I guess for you guys it's complicated to read, but it's minutes we are reading on the, so it's from 0 to 140 minutes, and already at 10 minutes we observe this increase of ATP in the medium. So meaning that no-breath inside action is very quick. And we thought it's interesting to notice that because of our recommendation. I don't know if all of you are familiar with the good way of use of chitosan for antimicrobial action, but what we would recommend is to, actually it's a powder, so, and it's insoluble. That's one of the specifications that preparations have to comply with. Chitosan is a processing aid, not an additive, and it's insoluble, so experts have established that it's eliminated when we have a rack-off. So, but what we recommend to get this biological effect and physical effect is to add it to the must or the wine you want to treat, homogenize perfectly, and let it settle for 7-10 days minimum. Can be more. This is some point we can discuss. And then rack off, especially if it's a curative treatment. So people were telling us, some winemakers were concerned because, well, in 10 days, many things can happen. This means that no breath inside is not active quickly. You mean that it's not active quickly? And so we made some experiments, and because also out there, some are talking about activating chitosan. This is something that sounded strange to us. Why activating chitosan? Our product is active. We've selected the raw material that has the good properties, and it is active. And it is active quickly. Doesn't need to be activated, at least the raw materials we are playing with. And so in these experiments, what we show here, you have 3 groups of bars, time 0, 5 days, 20 days, and there is a follow-up of the populations either on the control, so these are the 2 first bars, and then you have the second 2 bars are the treatment with no breath inside, and the 2 last one with a so-called activated chitosan preparation. You have counting. The first bar for each series corresponds to classical quantitative PCR, and the second bar corresponds to Pmax PCR, because you know with classical quantitative PCR you might have the occurrence of false positive results. Pmax PCR enables to make sure that you count living cells, meaning that there is an assistance with a molecule that will bind with the dead cells and make that we don't count dead cells. And so what you can see interestingly here is that at day 5, we already have eliminated or killed the Brettanomyces, and not in a less efficient way, even on the contrary, because it's faster than the so-called activated chitosan. After 20 days, okay, activated chitosan or ours is as efficient. So challenged by the techniques, we have also collaborated with a team in Bordeaux and on flow cytometry, because this is an interesting tool to count microorganisms. It's also a matter, as Nicolas was saying, what technique, what level of population am I counting? But there are serious advances in flow cytometry. And we were also, we have always investigated on the efficiency of our chitosan on different strains, different levels of populations, different physiological status. But at last, we could make it to study it at lab scale, to have standardized conditions. And so in that case, we would work with a rat population that is either In this little table, at the bottom, the right bottom of the slide, you can see there is a first column where we are talking about a low level of population, 10 to the 3 cells per milliliter. On the right is 10 to the 5. So we are playing with this level of population and with the physiological status of the cells. So meaning either growth phase or stationary phase. Because we know that depending on the phase of the growth of the microorganisms, the cell wall might be of a different kind, and so the interaction with chitosan might be different. So we are playing around this. If we look at the results, you have 4 lots of bars, T0, 10 days, 38 days, and 73 days. The greenish bars are referring to the control. So you can see the evolution of the breadth, whether the population is medium or high, whether it's in growth phase or in stationary phase. You can see that the population stabilizes and then increases after 2 months and a half in the control untreated. All the bars with warm color, this is how we call it in French, maybe sounds weird in English, but meaning red, orange, yellow color. You have the modalities that were treated with no breath inside at 4 grams per hectoliter. And so what you can see is that you have T0 and day 10, that is just before rack-off. You can see that we have a strong decrease of the populations before rack-off if we conduct flow cytometry. After rack-off, we cannot count any Brettanomyces anymore. And if we go to the point at 2 months and a half, the control is higher, but in the modalities treated with no breath inside, we see a bloom of Brettanomyces, but to a reasonable level of population that can still be controlled. Here it's important to say that we are in the conditions of very low SO2. But most interestingly, and especially for the enologists in this Bordeaux area where we are working with, friends of our friends, of some friends that had an issue, and these enologists were really comforted to see that, because it means that they were not panicked because we see a bloom of breath afterwards. Because if you monitor your populations, then you can take action and treat again or do another kind of operation. So it's still time to intervene on your process. So here what you see is the volatile phenols. So we see the green bars that are increasing all along and reaching very high levels of 4EP, 4EG at 2 months and a half, whether you cannot see or very low level of red, orange, and yellow colors on the bar. So this made the enologists with whom we work very comfortable, because they say, at least I can prevent from volatile phenol production. And even if I get those 10 to the 2, 10 to the 3 cells, I know now, because I monitor my populations, I can take action and fix it.
Do you have 4EP and 4EG results for the same table?
Yes, we have both data. Yeah. Here is the sum of them. That's just for illustration. But we have data, the details that were shared. It's just to make it synthetic. Well, another challenge for us, for winemakers, is that it's more and more reported, maybe part of your observations, that there are the occurrence of strains that exhibit tolerance or resistance to SO2, which is the privileged tool for winemakers to control microbiologically. So there is this study on the left-hand side. You have this extract of a publication from AWRI, illustrating actually the level of resistance to SO2 of different isolates that were collected in blue in between years 2000 and 2004, in red in between 2010 and 2014, and in green 2016, 2017. What do you observe here? We observe an increase of the resistance to SO2 from 0.5, 0.6 in years 2000 till reaching 1 milligram per liter of active SO2. You know what that means impossible to manage under conditions of red winemaking. So this means an increase in the tolerance or resistance to SO2. There is another study where several teams in Germany, Australia, and Bordeaux were working together, gathering isolates, collecting isolates of Brettanomyces from wine, beer, kombucha, distillates, biofuels, because Brett is present in all these matrices. So they gathered almost 1,500 isolates, strains of Brettanomyces. They characterized them according to phylogenetic groups. And so in another study, this Avramova, from which the paper is, the result is taken from one of the paper, studied the tolerance or resistance to SO2, working on 143 isolates, and they pay attention to take bread from the different phylogenetic groups, assuming that genotype could condition phenotypes when it comes to resistance to SO2. So this means we have a good representative population of strains that were studied and that are likely to represent what we have out there in our cellars. And the diagram here, the circle, indicate the different type of resistance to SO2. In red, you have the proportion of strains among these 143 that are resistant to SO2. In yellow, there are the tolerant, and in gray, you have the sensitive. Well, you can see the empty glasses or the full one, but it's something really worrying, because the final statistic is that if we consider all the isolates from the different matrices, 36% of them are tolerant or resistant to SO2. And if we come to the strains coming from the wine, it's even higher. It's 43%, 46 strains out of 107. So it's really interesting and important to consider other tools than SO2, especially in that condition, because we induce a kind of selection. And at the scale of a winery, we can easily imagine that if a strain remains, it's the one that is the best adapted to your winemaking conditions. So here we made an experiment working on three different strains of Brettanomyces that were taken out of this collection of the published paper I just commented. And within our lab in Toulouse, we put a challenge test in place where we would standardize the condition on a grape juice, which we inoculate with the Brettanomyces population. On this first chart, you have a strain, BRT1, that is resistant, tolerant to SO2. It was clearly characterized. If it's alone in the medium, you can see how it grows at the scale of alcoholic fermentation time. When we inoculate with a selected Saccharomyces cerevisiae, where the most ancient biocontrol tool that since the 70s in the last century is bioprotection, we can call it, you can see we have a kind of decrease control of the population, but it's not enough. We finish with 10 to the 5 cells. In the case of inoculating with Saccharomyces cerevisiae and adding sulfites, we don't do better, but no Brett inside, yes, has an impact on this strain, which is resistant to SO2, and so we observe a decline of an elimination of the population. I don't detail the figures for the two other strains that were taken out of the sensitive group, and you can see always the same strain. Sometimes you have 10 cells or less than 100 cells at the end of fermentation, but we also followed those wines after fermentation, after centrifugating the treatments, and follow them up to 3 months. And on the treatment with no Brett inside, we didn't observe a bloom in our lab scale condition, a regrowth of Brettanomyces cells. So this is to conclude on this part related to Brettanomyces and no Brett inside. So remember, we can now be sure that it's important to use it in the good way to ensure both biological and physical action. It is effective according to the studies we made internally, but also with many academic researchers in each of these teams where we put no Brett inside. We observe this efficiency, different strains, different levels of population, different physiological states. And if we come to the cellar, actually, beside the our recommendation, best recommendation, I detailed that already. If you are in a curative treatment, it's really important to, beside this great homogenization, to wait 10 days and rack off. If you want to use it in a prophylactic purpose, preventive way, of course you can leave it in contact. We had experiences with that for people who want to treat for a preventive objective when they rack their wine to barrels. They don't have a specific contamination they can identify on their wine, but they want to be quiet for aging only, and so you can imagine to use.
If you are using it in that more preventative care measure, would you use less if it's not in a curative measure?
It's a good point, actually, and we have friends now that give us trust in the fact that, friends in Rioja, in Bordeaux, in Tuscany, where actually this is something we easily now, we were not comfortable at first, 10 years ago, but now we get more and more of these feedbacks, actually, and you can play around 2 grams per hectoliter when you rack your wine to barrel. Of course, you have to monitor your contaminations, and so this leaves also some opportunities to treat later if you have a bloom.
And then on a follow-up with that, if it is being used as more of a preventative measure, does it need to be stirred every so often to re-put it into?
This is also the question we get most often. We made a study on that. We have a publication on that topic we can share with you. So, but you may be not happy with the, the results of these studies, because actually we couldn't clearly state if it's important to steer or not to steer. We work on in Tuscany with a lab in winery scale, in barrels, on a Sangiovese matrix, on a Merlot matrix, on 2 vintages. So of course it's not exhaustive, but we couldn't make clear, because in one case it was just the classic protocol of the winery, and the other case we would steer every 2 weeks to put the chitosan in contact with the whole volume. But at the end of the day, after 9 months, we couldn't see a difference in between steering or not steering. We could see the effect of chitosan to avoid the bloom of Brett at springtime, because on the control, that's where Brett grew at springtime with the elevation of temperature. But steering or not steering. So we say to winemakers, do not change your protocol. Keep it as such. If you see an increase of your population, it's worth resuspending and control again.
Some winemakers might decide not to use filtration prior to bottling, even though they know they have a Brett in the wine. And they might use Velcorin, for example.
Mm-hmm.
What about if you would use chitosan before bottling?
Well, chitosan is insoluble, so you would need to apply it and rack off and before bottling. But there could be some residual cells, depending on the method of control that you use. So,
it's not 100% in a sense.
Yeah, we never say, either for no Brett inside or Bacillus or Alliance, we cannot say it's the fatal tool that will eradicate.
You would use it or wait for the period of time.
Mm-hmm.
There is still possibility.
It's a tool to reduce population, to control, to keep them in check, as Nicola was saying, so to manage them, exactly. So it's not the tool for pre-bottling treatment to make sure that no cell will be in the bottle.
Question follow-up, similar to both questions.
Yes, please.
So you say the population is reduced just before racking, but how are you sampling that? Are you stirring to get the full picture, or are you looking at just the wine and maybe all the Brett is down in the bottom?
Yeah, we did. Didn't have the time to present all the details of this study, but we'll publish that soon, shortly. We made sampling at different heights of our containers, actually, and top, medium, bottom, close to the lees, so that we observed this gradient in the concentration of the cells. So there is the sedimentation effect, but yeah, we can manage this way. We also made duplicates, aliquots, where we would sacrifice one of the treatment. You can, it was a lot of samples, but yeah, it's a good point. Okay, so we go to bacteria, our second target, lactic acid bacteria and acetic acid bacteria with Bacillus. So it's not new neither, so you might know this tool. The particularity is that it's combining chitosan and chitin glucan. So yeah, the question addressed by the colleagues at first that time and still is that why couldn't we use no breath inside to struggle lactic acid and acetic acid bacteria? Because we found out that there were some other chitosan, and that by combining with chitin glucan, it can be even more efficient against those little microorganisms. Bacteria is smaller than yeast. It's not forcibly the same timing of treatment. And so it was really clear that by combining this specific chitosan with chitin glucan, we increase this sedimentation ability of the tool to reach a higher reduction of lactic acid and acetic acid bacteria population. So we propose Bacillus. This part will be a bit shorter. If you have questions, you can raise them. But so there are two main objectives: malolactic fermentation control. In the case you want to delay it or you want to avoid it, this is a potential alternative to lysozyme, to SO2, of course, or to a complementary tool to decrease your SO2 addition in this specific application, white and rosé, of course, when it's not desired. And in red wines, it's also a good option if you want to stay quiet for a microoxygenation or a time for your color to stabilize. If you want to delay malolactic fermentation, it's a good tool to control bacteria. The other big area of application is the stabilization of the wine after malolactic fermentation to reduce bacteria population. And so this is what is illustrated in the following slides, actually, where it's a work that we did with IFV in Burgundy on a Pinot Noir. The four first bar on the left are related to the Burgundy Pinot Noir matrix, pH 3.4. And this same matrix was adjusted in pH to mimic some more warm climate condition, pH 3.8 on the right-hand side of the graph. And so you have the percentage of reduction of population of lactic acid bacteria that is observed under control, that is the light gray bar. You have on the right in dark red and in pink the effect of SO2. First at high dosage and then at medium dosage. So you can see, of course, SO2 is the best performing tool at rather lower pH, but Bacillus is not doing bad. It's the green bar, and we have an impact that is even higher than the medium dosage of SO2, 15 milligrams per liter of SO2. And when it comes to more cool climate conditions, pH 3.8, Bacillus is as efficient as the full dosage of SO2 to reduce lactic acid bacteria population. Having said that, this was very standardized conditions, but we also come to the field with the team of my friend Pepe and the colleagues, the friends everywhere that have issue with spoilage bacteria. You have several set of experiments that were done at a winery scale in Argentina, in Spain, and in France. And you can see how it illustrates the reduction of the indigenous flora. So it was really the spontaneous flora that was there, maybe lactobacillus of different kind, pediococcus. And so you can see that in any cases, we have a reduction of population. Coming back to your question, we don't affirm that it eliminates 100%. Sometimes yes, sometimes no. And for this case, we have a decrease by 2 to 3 logs generally of the contaminants. Lactic acid bacteria and also acetic acid bacteria. As you can see here, it's interesting to see the last set of trial on the Merlot. Actually, there was no SO2 added in this process, and so we recommended to treat with 50 grams per hectoliter. And so we could see the reduction of lactic acid bacteria and acetic acid bacteria. Might not seem very important here, but when we consider the volatile acidity on the resulting wine, it makes the difference. You can see here easily the comparison between control and treated with Bacillus. And other experiences closer to you here in North America, also a lab scale experiment at the top illustrating at lab scale the impact on different kinds of bacteria, from acetic to lactobacillus, pediococcus, enococcus. So you see again this reduction of populations. And on the graph at the bottom, it's the illustration on winery experience here in Napa. So I was not here because I told you it's my first time here, but some friends were here to take care and share these trials. So last but not least, we have something new to share with you and to introduce you to complete the toolbox. Again, you understood our credo, our way of considering chitosan as a tool. It's not one formulation for anything. Here, what we were looking for is a tool we could offer to winemakers to treat their must early in the process, to prevent their wine from spoilage, but early in the process. So this preparation, Alliance, is really made for an application at the beginning of fermentation or during fermentation, or in case of stuck ferment, if you want to restart, because it's a tool to clean and sanitize the medium. And we are not talking only about decreasing the microbial load, whatever it is, because you will see there is a broader spectra of action. It's a preparation that can act against lactic acid, acetic acid, bacteria, but also spoilage yeast. But not only that, it's combining chitosan with specific yeast cell walls that have important detoxifying properties. So you know with L'Allemand, we are microorganisms producers. I'm more on the dark side, as Nicola said in her introduction. So I play around yeast derivatives, and yeast cell walls are being one of them. And so we have a bunch of different strains we are testing, and we have here really a specific wine yeast strain that was interesting for the capacity of its cell wall to sorb toxic compounds, namely fatty acids. That's what is illustrated here in this first trial that was run at lab scale. You have a fermentation that is stuck on the control. It's the dark blue bar. You can see residual sugar, 14 grams per liter. You can see how Alliance added at 30 grams at the beginning of the process can manage to avoid this stuck fermentation. We have a complete fermentation. And why? And this is why we choose Alliance, is because we have a decrease of the content in short and medium chain fatty acids. When the yeast struggles toward the end of fermentation, it produces this short and medium chain fatty acid that compromises even more its work till the complete fermentation. And so in that case, even added at the beginning of fermentation, we could show that it's still interesting to eliminate these short and medium chain fatty acids. In another trial here on Merlot at lab scale, also it's what we call crash test conditions, because we mimic an exogenous contamination with lactic acid bacteria. So on the graph on your left, you have the evolution of the lactic acid bacteria population in 3 conditions, the red line being the must that has not been inoculated with bacteria. Oof, we worked well. There is no increase in the lactic acid bacteria population. It's our negative control. Then you have in the orange bar, you have the control that, yes, was contaminated with lactic acid bacteria. Lactic acid bacteria grow, as you can see. And on the right, you can see that is the only treatment that ran through malolactic fermentation with the decrease of malic acid. And interestingly, the blue line indicates the effect of Alliance. And you can see even at 10 grams per hectoliter, in that case, it manages to eliminate lactic acid bacteria and prevent from early malolactic fermentation. On this same trial, in parallel, we were following the kinetics of alcoholic fermentation. And here what we show is the concentration, the consumed sugar. So the higher, the more the fermentation is complete. On your left, you have the 2 treatments with Alliance, 30 grams or 10 grams per hectoliter. You can see how it helped the yeast to go further, to consume more sugar. On your right, you have the first lower bar that is the same control inoculated with lactic acid bacteria, but no Alliance. So you can see that in this case, the presence of lactic acid bacteria has annoyed the yeast so that the fermentation is not complete at all. And on the right, you have this same control with without lactic acid bacteria. So if you compare the last bar and just the previous one, you can see how the presence of lactic acid bacteria compromise the fermentation. So to complete on the elimination, the detoxification impact of Allianz, here is a trial done in Galicia, well, on a must of Albarino coming from Pepe's country, where actually it was taken because, you know, these grapes, the winemakers struggle with a humid climate and so on, and so they have some treatments to run on the wineries, and so there can be some pesticide residues at the time of the harvest, and sometimes fermentation are complicated, and also there are some spoilage pressure, spoilage microbes pressure. And so they took this must of Albarino with not a high clarification level, 319 NTU. They divided it in 2 lots, a control and one treated with Allianz. It was settled at cold temperature. Maybe my time is going to finish, that's why. So 44 hours, 10 degrees C. And after this time of cold stabilization, the colleagues would measure different microbes present out there by RT-PCR and also the pesticides residues. And a bit further on in the process, 120 hours later. What could we observe on this trial is that, yes, there are spoilage out there, 610 to the 4 on the control. On your left, it's at 24 hours, so it's already a kind of high population. You can see how Allianz managed to decrease it by 52%. But most interestingly, we can see that after 120 hours, we have a growth of this Enseniaspora uvarum, this spoilage yeast, whereas Allianz enabled to control this spoilage yeast. And coming to the other guys that were present out there, the smaller one, Lactobacillus, you can see on your left, you can see the same trend, a reduction by 100% of the Lactobacillus brevis population already at 24 hours. It's maintained at 120 hours. Of course, fermentation has started, so on the control, it's repressed. If you go to Acetobacter, the acetic acid bacteria, you can see also a strong trend of Allianz to manage to decrease, eliminate these bad guys. And coming to the pesticide residues, here are the ones, the compounds that were found in this specific Albarino must. And so you have the bars on the must at T0, the last bar being the sum of the pesticide residues. And you can see how it evolves. On the control, it's the light gray bar, 24 hours. And on Allianz treatment, also 24 hours, you can see that there is a decrease in this pesticide residues content by 15 to 30%, which is likely to help the yeast to ferment in good condition. And so this is what I wanted to share with you on Allianz specifically. And coming to my conclusion, I want to add this parenthesis about the good compatibility with our other living biosolutions for controlling antimicrobial risk, but not only. Pepe commented about bioacidification, protection against oxidations. So you have at the bottom of the slide our different non-Saccharomyces that can be good to combine with chitosan solutions without any problem of compatibility. This is something we checked first at lab scale, but also now that it was implemented at winery scale. And regarding malolactic fermentation, we are lucky to introduce ML prime. We can make sure that there is no incompatibility. So could be a good strategy to imagine in early stages combining ML prime and Allianz, for instance, for best control of the risk. Of course, in co-inoculation, we would really favor ML prime. We observe some inhibition of our Enococcus any solutions. So this is just how you can best choose your allied tools for your winemaking process. Allianz at the early stages with a broader spectra of action, spoilage yeast and bacteria. If you target bread specifically, no bread inside. And for lactic acid and acetic acid bacteria, Bactriless would be still the best match. I thank you for your attention. If there are any questions, you're more than welcome. Thank you.